Okay, thank you, Manjuna. So good evening to all the participants. I take it as an immense pleasure to introduce to you all to about Dr. Bipin Singh. Sir is Assistant Professor, Bioinformatics Department, Center for Life Sciences, Mahindra University, Telangana. So Sir has been served as an Assistant Professor from the Department of Applied Sciences, BML, Munjal University, Haryana, from 2017 till 2021, and has continued in the same
Bipin Anna,
profession.
namaste.
And continued in the same profession as an assistant professor in bioinformatics, Department of Biotechnology from.
We request all the participants to be muted.
Please remove that participant, Mukesh.
Okay, ma'am.
Greater Noida, Uttar Pradesh. And currently, Sir is holding the position of Assistant Professor Bioinformatics, Center for Life Sciences, Mahindra University, Telangana, till date. Coming to Sir's educational background, Sir has completed his master's in bioinformatics with the distinction from University of Allahabad, Central University of Allahabad, Uttar Pradesh, 2008, and also has been awarded PhD in bioinformatics from IIT Hyderabad in 2017. And Sir's dissertation topic is understanding the molecular basis of thermostability and activity of Bacillus subtilis lipase and its mutants. Sir has received a lot of honors and awards to his credit, which includes Drug Discovery Hackathon for COVID-19, GOI, MHRD, CSAR, on the theme or the topic, developing a regression-based model for screening compounds with 3CL-Pro inhibitory activity in the year 2021. Sir has received Institute Postdoctoral Fellowship from IIT Delhi in 2018 and also from Graphics Processing Unit Hardware Grant NVIDIA Corporation in 2017. Sir has been awarded a lot of awards or prizes as in the best poster presentation and in the best oral presentations. Sir's main area of research interest and expertise is in computational drug discovery and AI for bioinformatics and healthcare. Sir has been funded with two different projects. One project has been funded worth rupees 10 lakhs from iHub, that is Technology Innovation Hub, DST, Department of Science and Technology, IIT Hyderabad, for the title project, development of an AI-based platform for glaucoma detection tailored to work with handheld fundus cameras in real-life settings. The second project is being funded with an amount of rupees 4.7 lakhs as a seed grant from Mahindra University for the project title Validation and Re-engineering of Peptides Derived from Monoclonal Antibodies for Ocular Tuberculosis. Sir has done a lot of publications to his credit in reputed journals, and the number of citations to his credits include 284 with an H index of 6 and I-10 index of 3. Sir has been contributing even for preprints as well as for conference publications and also for book chapters, and also has been a main invited lecture person or a resource person in various conferences and also is presented in different conferences. Sir has been a course instructor. Main area of courses include bioinformatics, algorithms in bioinformatics, computational drug discovery, on biostatistics, Python and BioPython, AI and data science for biotechnology, data management and engineering, and public health informatics. Sir has completed a lot of professional certification courses also, which includes faculty development program in advanced techniques in life sciences, University of Delhi, and AICTE Training and Learning Academy FDPs on artificial intelligence, computer science and biology by National Institute of Technology, Arunachal Pradesh, and applied machine learning by IIT Punjab, then an FDP on computational synthetic and system biology by Indian Institute of Technology, Guwahati. Coursera certification courses also has been completed by Sir. So coming to Sir's professional affiliations include convener for the Regional Student Group, International Society for Computational Biology, Hyderabad chapter. Sir has served as a member for International Society for Computational Biology, USA, has been a member for American Peptide Society, USA, a steering committee member for India RICS Preprints Repository for India, has been a mentor for Asia Pacific Bioinformatics Network Student Ambassador Programs. Sir has got a lot of science communications also to his credit. And coming to the professional services, Sir has been a reviewer for a lot of journals. That includes PeerJ, Helion, Biomacromolecules, Journal of Molecular Modeling, International Journal of Computational Biology and Drug Design, International Journal of Immunopathology and Pharmacology, Current Computer-Aided Drug Design, and Letters in Drug Design and Discovery. Sir has also been served as a convener for an international workshop on AI in healthcare, discovery to delivery from 16th to 20th of August 2024. Sir has also have a PhD research scholars under him who is still guiding 3 students who are pursuing PhD under him. And also Sir is performing his thesis supervision for 2 of the BTech computer science students. So thank you so much, sir, for accepting our invitation and joining us for today. And sir is here to deliver a lecture on the very interesting and upcoming important topic, applications of artificial intelligence and machine learning in drug discovery and protein design. Thank you, sir. And now the session is open to you, sir.
Thank you very much for this long introduction. So I will share my slides and we can confirm whether. So can you hear me loud and clear?
Yes, sir. Absolutely clear, sir.
Okay, good. So
PPT is visible, sir. You can put on slide mode.
yeah.
Yes, sir.
So welcome to all the students and thank you for inviting me for giving this guest lecture on this topic. So I'm going to talk a lot of things today, and 2 hours is a very short time, but what we'll try to try that as much as knowledge I can impart in simple and easy manner. And so that if you have more interest in these areas, there are ample of resources available on the internet, or you can also contact me later if you do not thoroughly understand some of these things, because 2 hours is a short time to explain this topic, but I'll try my best to deliver. So this topic of my presentation is AI and ML in drug discovery and protein design. And I hope all of you are using AI and machine learning in some of other way, may not be in sciences, but in your daily life. So you must be all aware of what is going on in the area of AI and machine learning. So I'm going to touch upon how AI and machine learning is used for drug discovery and protein design. And as far as I know, all of you are from undergraduate and maybe from biological sciences or other domain, but I'll try to explain what I mean by application of AI and ML in drug discovery. So I'm going to talk about drug discovery and protein design specifically, and how AI and machine learning technique have been used in these 2 areas. And the right hand side of my first slide, I'm showing what is actually AI. So I'm going to use this term very often. So AI is a very broad term. There could be multiple different kinds of techniques which comes under AI. It could be a simple machine learning or more complex, which is called deep learning. So you don't have to bother about when I'm using AI, deep learning, and machine learning interchangeably during my talk. So I may mean that one of these methods, how this have been used. Okay. So as far as importance of this area, AI is touching upon every aspect of our life, and likewise, bioinformatics and drug discovery is no different. And if you see this landscape, uh, though the market size which is showing here, so market size means that how much industry is using these techniques for different kind of services and products. Okay. So the market size for AI application bioinformatics is huge. And that area which I'm going to talk about is the second on the right hand side, with two bars you can see chemoinformatics and drug design. These are the broad area which comes or which closely aligns with the topic of the talk today. So these are the two major areas where AI finds an application when we talk about life sciences. One is genomics, which is again the theme of this value-added course. But I'm not going to talk about genomics, but I'm going to talk about more drug discovery and protein design, which falls into this area. So application-wise, this is also a very key area where a lot of companies, it could be pharma companies or lots of startups which are working in this area in innovative product design and also imparting services to large companies. And therefore, the market size is expected to rise to a great extent in future. So it would be good for students, even if you're not learning AI and machine learning, but some aspect of this you can learn whatever area you're interested in, whether you're interested in genomics or if you're interested in broadly in proteomics or drug discovery. So try to learn something related to AI and machine learning that is going to help you in future. So before I begin my talk, so the area which we are going to discuss today is very, very relevant. If you see, if you have seen a couple of months back and noted that this Nobel Prize which was awarded in chemistry. So the Chemistry Nobel Prize was awarded to three scientists. So on the right hand side, this David Baker have been awarded Nobel Prize in chemistry, but not for a pure chemistry problem, but the problem of computational protein design, which is today I'm going to talk about. And it closely resembles with the problem of protein structure prediction. So I will tell you what actually I mean by protein structure prediction, and computational protein design in later part of my talk. But this area is very recent, and people are working in this area for a long time. But now more and more people are going into this area and trying to work on different testing problems. So this Nobel Prize is also signifies that the importance of this area in future. And likewise, the Nobel Prize for Physics was also slightly different into 2024, which was not awarded to a core physics problem, but it was awarded to two scientists who have discovered how deep learning methods, which is also called artificial neural networks methods, works. So these two discoveries are using the discovery of protein structure prediction by these two guys, which are from Google DeepMind, actually use the technique developed by these two scientists. So it tells you that how world is going through and what you can expect in future, whether you're working in pure sciences like chemistry and physics. More and more people and more organizations are going to use these methods like AI and machine learning for different kind of research or product development or even designing new therapies. And again, it would be good that if you have any question or queries, you can unmute and ask your question after introducing yourself. And so that you don't keep your doubts with yourself. And if you wanted to clarify anything, you can stop me in between and ask your question. Okay, so we can make it more informal. So when we talk about AI and machine learning in drug discovery, I mean, I hope all of you know that what we mean by drug. So drugs, like any kind of medicine you take in daily life, these are not something very new to us, right? So we are taking drugs since centuries, and people are working on discovering new drugs from centuries. Then question comes, what is the use of AI then? People were discovering drugs since centuries, so why suddenly we feel that AI can do something new in the drug discovery? I'll explain why it is required. So, but here what I'm trying to emphasize that AI is not something very theoretical or only in textbook. Uh, the drug designed by AI and machine learning are actually reaching to the stage of clinical trials. And I hope you know that clinic. So any drug, if some organization or company has to market, they need to qualify all the phases of clinical trial, showing its safety and efficacy.
టైం
So
వేస్ట్
many of
చేస్తున్నారా
the
మీరు?
yeah. So if any question, yeah, please unmute and ask for. So here, what I'm showing that AI designed drug, which is coded by this name, have been granted clearance for cancer clinical trials, and that shows that AI designed molecules are not completely theoreticals. And likewise, you have multiple other such reports where a molecule designed
Good
by
night.
AI has been reached to clinical trial stage and showed its efficacy against various complex diseases. And then I'm going to talk about protein design. So all of you know protein. Proteins are of different kinds, and whether it's of structural protein, non-structural protein, it's occur everywhere in nature, and 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 protein finds its place in multiple industries. So protein can be a target of drug, and protein can also work as a therapeutics. So protein is very important, but when we want a design protein from the scratch, it's a very difficult problem. And it's this structure which is showing here is a protein bound to a DNA. And suppose we want to design a protein which can target a DNA which has certain mutation, like in cancer, it is not very simple problem. So if as a biologist or biochemist, I want to design a protein molecule which should bind to DNA, doing this from the scratch can take a lot of time. So what advancement happened in AI which can do this thing in very easy manner? I'm going to talk about that aspect too. And then if you understand the role of AI and machine learning in drug discovery or protein design, it has a lot of practical application, like accelerating drug discovery again. So these are the case study which people have used this kind of information to solve problem like accelerating drug discovery against malaria, discovering novel drugs against drug resistance pathogens. So the problem of antibiotic resistance is well known, right? So we are taking antibiotics since long time. But now what we see that the same antibiotics are not very effective in various disease cases. And if the patients are critically ill, and if you have a limited kind of antibiotic, and if they're not responding to those in antibiotic, the situation becomes very critical. So how novel antibiotic design can use these technique of AI and machine learning, I'm going to give you a case study about that. And likewise, there are so many problems, and if you're interested in any kind of problem, whether it's genomics, proteomics, or drug discovery, AI and machine learning found some application for that. So today I'm going to touch upon this part, which is drug discovery and protein folding and protein design parts. I'm not going to talk about much about other applications. But AI on deep learning or machine learning, I've mentioned in the beginning, I'm going to use this term interchangeably multiple times. So AI and deep learning, how it can be used for drug discovery and protein design, that I'm going to discuss in this.
Please mute yourself, participants. Sorry, sir.
Yeah, no problem. So first question somebody can ask, okay, so can AI replace, I mean, why we need human if AI can do every job? The question is, and you all must be using some kind of AI. So if AI and machine learning can do everything, what is the need of human researcher? Anybody want to answer?
Participants, you can respond. Anyone?
The question is very simple. I'm discussing about AI can do this, that. If AI and machine learning can do every task, why we need human? Why we need human to discover drug? We can outsource for the drug discovery project to AI and machine learning models,
So,
right?
so AI is like a tool. It cannot replace the human thinking. That's one thing I find.
Okay. But have anyone have used ChatGPT here? You must have used ChatGPT for some of the other work. Anyone? Have you used ChatGPT?
You can raise your hands or give a thumbs up.
Yeah, there are a lot of hands. Yeah. Now we are facing a lot of problems, right? I mean, we give certain assignment to student, they go to ChatGPT and find out the answer, right? ChatGPT knows everything. So likewise, if we have a very accurate AI and ML model for drug discovery purpose, why we need a human researcher input? That is the question.
Well, so I think it's because we also need to cross check if it is right or wrong, because AI is a program to just look for results and they probably get a match, but that may not be, something that we are looking for. That is why there needs to be a human intervention.
Okay. Yes. So like we have seen many times, suppose we ask certain assignment problem, student go to AI and just type that question, and whatever answer AI gives, they sometimes blindly copy and submit that without checking the fact that AI is not perfect, right? I mean, it can do mistake like human, but human, we know, and many times human are aware of their mistake, but these AI system, they're not still aware what they're doing sometime, or they cannot explain what kind of error they're doing. Like ChatGPT, many times it's tried to create a knowledge which is factually wrong. Okay, because it have a problem, something called hallucination. When you don't know something, you try to create some imaginary output and dump it, and the user will realize that it's a correct information. So when you say AI for drug discovery, like this example which I'm showing here, so when AI design drug doesn't mean that you have a machine, you feed the data, and the drug will come as an output. So whenever we are going to use AI, that is in combination with the human researcher, but
Prince,
not.
please don't mute.
Please keep yourself on mute, participants.
Many students are listening songs side
I'm
by
sorry,
side. It
sir.
seems.
Extremely sorry, sir. Only
No,
today
no,
it's
no.
happening.
That happens. I know. So many students who might not be interested also sometimes joins.
Excuse me, sir.
So yeah,
Sir, can I just ask you one question? Like,
please.
how does this AI work? Like, I mean, AI is a adaptive engine. Like, it will be feeding on the previous results to
Correct.
give a prediction of further results.
Correct.
But how does it work actually? Like, as of Google, we know quantum engine it uses to for quantum analysis and all. But how does AI work? And
Uh, so AI is a very broad term, right? Suppose I go to the example of this deep learning kind of network, right? So you have certain input, it learns some features, and then try to predict the output. Like and for any AI system, we generally call it training, right? So like ChatGPT, how ChatGPT knows everything, right? It must be trained on all kind of input, right? So ChatGPT have been trained on billions and of data set, which might sometime open source and closed source. So you feed everything, whatever knowledge exists on Earth, and then it try to learn all the features which are present in that data set. And then if it has learned everything from that data set, ultimately whatever you're going to ask, it might if it has learned everything in very accurately, then it might going to synthesize the same knowledge if the question is asked accurately. So think here, suppose a deep learning model. So these inputs would be, suppose, a different kind of data set. Then we need to encode that data set so that the machine understands. Like in the case of drugs, so suppose we have a drug molecule, then it will have a chemical structure. And then chemical structure will have different kinds of atoms, different kinds of bonds, different kinds of other derived features. So it will try to extract all kind of information which it can extract. And then it try to find out if given this all the possible features it has extracted, how it correlate with certain output like antibiotic. If I suppose I want to build an AI model for antibiotic discovery, what I'm going to do is whatever antibiotic kind of molecule exists on in different databases, I'm going to feed that data set as an input, then trying to calculate all the features which can be calculated from data set. And then as output, I'll find out, okay, because it have learned all the antibiotic feature, now it can synthesize something like that. Or given a molecule as an input, it can tell whether that molecule have antibiotic feature or not. So ultimately, it's all about learning from the data set, using different kind of features. In the conventional machine learning model, we use to handcraft feature, but when we use AI, we don't handcraft feature. It tries to extract all possible feature which can be extracted from a given data set. I'll show you further example later on, then it will become clear. So the question is no, AI cannot replace human. AI is to assist human. So like in drug discovery, those drugs are not discovered only through AI models. So human must be in the loop to fact check what AI is doing, ultimately teach them how to discover something novel. So in general, when we talk AI in machine learning, there are two kinds of methods which are used, something called supervised learning and unsupervised learning. So this is mostly people try to use it. So supervised learning, like you try to have, you must have a label data for that. Label data in the sense, suppose I have an antibiotic, and I know that this particular molecule has antibiotic nature. And the reason for this antibiotic nature is suppose some labeled features. So many times for supervised learning, you need label data. Label data means you have information about the output. If it is suppose a chemical molecule, you know that the chemical structure and the chemical structure, whether it's antibiotic or not, it is already labeled. Then you try to learn those how the output that is antibiotic positive or antibiotic negative is correlated with the input structure feature. So this is called supervised learning, where you supervise or you try to you try to you try to calculate feature using some already existing knowledge, and then try to predict what could be the potential outcome. So supervised methods are used since long time for different kinds of purpose, like listed here. It can be used for different kinds of application in drug discovery, like you can design molecule from the scratch. You can predict whether a molecule is going to be highly active or low active. You can use for classification purpose, predicting the adverse reaction, and so on and so forth. There are something called unsupervised methods. Many times we may not have a label. Suppose you take some chemist gave you 100 molecule structure, and he has not told that which molecule can be antibiotic. He said that I don't know which molecule can be antibiotic. Maybe you can use some method to tell me that. So to tell about that. So then what you do that, because the labels are not available. So what you do that, you only have the input structure available. So you apply some technique to cluster them into different classes. So clustering or dimensionality detection are these kind of methods in machine learning or AI, where the output labels are not available. Therefore, we try to cluster them using different kinds of features. Like for example, I have hundreds of antibiotics. I will calculate molecular weight, I will calculate solubility, surface area, electronegativity, everything, and try to see that whether using these property I can cluster these molecules. And if I find multiple cluster, suppose for example, if I can draw here. What I mean, like, suppose you find multiple cluster like this, right? So you have a cluster, two cluster, and then you say that this is a cluster where solubility, electronegativity, and other properties are different, and this is another cluster which have different solubility, electronegativity. So maybe this cluster could be for antibiotic molecule, and this cluster could be non-antibiotic. So you don't have that label present before, but you still can classify by using some technique. So these kind of method, which does not require the labels, are classified as unsupervised learning methods. And the supervised methods often require labels. Label means I know what is antibiotic, what is non-antibiotic kind of molecule. I'll try to predict what kind of feature make this molecule antibiotic and what kind of feature might make a molecule non-antibiotic type. Okay, so this kind of classification we can done using supervised methods. So coming to our original agenda. So our agenda today is the application of AI, right? So drug discovery is a long process. So here I'm showing that this whole drug discovery process often take, you can count the years. So generally suppose 1 to 3 years for discovery development. Preclinical development is before launching a clinical trial, you want to test the safety and efficacy of the drugs in different kinds of animal, mostly mammalian systems. And then you do a clinical trial, which can span from 5 to 7 years. So the overall drug discovery process is a very lengthy process. It may take 7 to 10 years and maybe beyond that. So the question comes, many times we don't have this amount of time available, right? Like we saw in the COVID. So COVID came. So if we wait for 10 years to discover COVID drugs or vaccine, obviously it's impractical to imagine, right? So many times what we need is how we can reduce this time. Okay. So AI came into picture in drug discovery only to reduce the time and money. If the time and money could not be saved using AI application, there is no use of AI in drug discovery. So if you want to use AI in drug discovery, our objective is to reduce this time. Can we discover drug in just 5 years? Okay. Or can we reduce this cost, which is often very high, which is listed here? It's the average money which pharma companies spent from discovery to final approval is sometimes cost billions of dollars. And if you can imagine, if something fails in the clinical trials, the pharma company is going to lose at least 1 to 1.5 billions of dollars, and therefore it would be lot of loss to them. Okay, and that is the reason why in India we do not have a lot of pharma company which discovers new drugs, because it's a very risky kind of business. The chances of failure is very high. Okay, because the drugs often fails in clinical trials. Okay, so the thing is, if we are spending a lot of money, can you reduce the money so that even if you face failure, you're not going to lose billions of dollars, slightly less than that? Or you don't have to wait for 10 years for discovering a drug. Can it be made in 5 years? That is the reason why AI and machine learning kind of methodology finds its application in drug discovery. Okay, the reason is not to replace human, but to reduce the human effort of discovering drug from the scratch to the final approval. So if you have not done any drug discovery course, I'll just explain in brief what we do here. So drug discovery starts from target identification. Like suppose a bacterial infection. So bacterial infection, suppose a bacteria is causing a respiratory infection. So if you want to discover a drug which is going to target a bacteria, so the first question comes that what you're going to target in bacteria. The bacteria is a whole organism, right? So whether you're going to target the bacterial cell wall, are you going to target some important proteins involved in ribosome production in bacteria, or some protein which are involved in cell wall biosynthesis? So the target discovery is the first step of any drug discovery process, right? So the question comes like, if you have hundreds of target in a bacteria, how are you going to select which target I should choose for my drug discovery project? Okay, so that kind of problem AI should able to solve. The question comes how? Because target validation, because if you're discovering antibiotic, you're not discovering it first time. Many people must have discovered antibiotic in the past for a similar kind of bacteria. So we have to look at which kind of strategy was very successful, which kind of target people have already validated. So that kind of strategy, AI can quickly tell you, right? I mean, you just scan through, okay, which kind of homologous bacteria exist, what kind of target has been used for targeting using different small molecule. And your target discovery task, which generally takes 1 to 3 years, can be reduced to max 2 to 3 months. So you're going to save at least 2.5 years in target discovery, which without using AI and machine learning, you're going to spend at least 3 years. So target discovery is the first step of any drug discovery process. And if you can use intelligently any AI and machine learning method to discover a potential drug target in quick time, that is going to save both time and money for pharma industry. Also, I'm going to talk about AI can be used for target discovery, which could be like which target you're going to consider for your drug discovery project. Once you have target you have validated, then comes into picture, okay, what kind of molecule you want to design. You want to design a drug which is a small molecule like antibiotic, or a large molecule like antibody or protein or a peptide. So different kind of so you have a target, but bullets can be different. So bullets can be of different size, and many times the bullets are decided based on the biology of the target. So you understand the biology of the target, accordingly you decide which bullet is going to kill that target. Okay, so the knowledge of both the target and the bullets need to be taken into account while discovering a drug. And this also have various AI methods which can do this process for you. And then ultimately, once you have finished this and you have a very robust result, then some experimentalists can validate whatever they have discovered in the lab, whether it's working fine or not. So overall process of target discovery, screening molecule, whether it's a small molecule or large molecule, can be done in a very quick time if you use different kinds of AI methods. And then a lot of money and time can be saved. So whatever money you have saved, maybe you can use for this preclinical testing, because any drug, it doesn't mean that you're applying AI, then you can bypass the testing on the animals or relevant mammalian models. So whatever molecule you have discovered, you have to ultimately test in a mammalian system or a cell-based system based on the complexity of disease or the complexity of the molecule. So once it shows a very good result in preclinical studies, and then you have very positive data about your drug through in vitro, in vivo testing, then you decide to take that drug to the clinical trials. Okay, I'm not going into more about clinical trial because that is another story. And once you are successful in clinical trial, then that drug can be given a FDA approval, and that can be marketed by that pharma company. So this whole process of discovery, which generally takes on an average 10 years, can be done quickly. If I can save a lot of time in target discovery, a lot of time in screening billions of molecules, and a lot of time even for clinical trials. So how AI can save, and AI is finding its application in all these domains. It is not possible to discuss all these aspects in 2 hours, but I'll try to show you a certain example how people are doing that. So the problem is very large. I'm just starting from this part. You have a target, suppose validated. Now you have billions of molecules. One way is that you test each and every molecule in your experimental lab. That is going to increase the cost. What I want is how I can reduce this cost by screening very less number of molecules and very quickly. And that is where the challenge is. So we have to find out in millions of such molecules which are there in our databases, how we can screen them very quickly and find my magic bullet, which can kill my target, which could be a bacteria or a virus. So in principle, we have multiple database, like you have a drug bank, we have Campbell, you have Zinc, which contains lot of chemical molecule, but experimentally it's not feasible to test all these molecules in the lab. Experimentalists can test hardly, I mean, they want to test least molecule so that they can save time and money, right? So if a pharma company comes to you that I have a target in a bacteria, I want to design a drug, but I don't want millions of molecules to be tested. I just want to test maximum 100, 200 molecules. So then you have to take some computational help. How are you going to screen this millions of molecules very efficiently? And if we have a good methodology for doing that, I can with confidence tell that, okay, you take this molecule, it is going to work in the lab. Okay, so that is our target. How to test minimum number of molecule to spend less money and less time. And this I want to automate using my computer. And generally, our laptops and these systems are not enough for that. We need more computational power and generally a high-end machine for doing this process. Okay, so how we can automate this process of screening molecule? Okay. So apart from that, I will come back to this slide later on, but so right now I'm going to discuss this part of AI in drug discovery. Like if you have millions of such molecule, how are you going to use certain AI technique to give me certain ranked list or certain curated list of molecule which can be tested quickly in the lab? So generally how it works is like you have this kind of databases, which name is like Campbell, PubChem, and DrugBank, which gives you a lot of such compounds and associated structures. So structures contains the information about activity of any molecule. Like you must have studied in class 12 chemistry, the feature of any chemical molecule depend upon its structure. Ultimately, the nature of molecule, whether it has rings, it has aliphatic chain or aromatic characters, all these different properties determines what kind of property a molecule is going to show. So the first task for applying AI in screening a potentially good molecules against a target, which could be bacteria, virus, is that you go to any of these databases, collect some compounds and their structures, and then try to formulate a strategy how you're going to pick up some key molecule which might be very efficacious against a particular bacteria and virus. So the first step is extraction of these compounds and their structure. And then there are softwares and tools which can calculate these properties for you. Like you have, suppose you have just doing a small project, you just take hundreds of such structure and then you calculate the chemical formula, molecular weight, and basic chemical properties like solubility, aromaticity, how many rotable bonds are present, what is the log P value. For such simple properties, there are software which can calculate for you. So what you get here is for hundreds of such structure, these molecule, which is in a Excel sheet or a tabular format. And this tabular format ultimately goes to any AI model to do some magic. Okay. So what showing here, I'm not without going into complexity. So from this structures, you can calculate a lot of features which many times is capturing the one-dimensional properties of this molecule and many times three-dimensional, because any molecule, it's not a 1D, it doesn't have a 1D kind of nature, right? So all the molecule, all the atoms exist in three-dimensional environment, or they have three-dimensional properties. So from the structure itself, there is a possibility that you can calculate all the features which is one-dimensional or two-dimensional, or they can also be encoded into some complex format like a graph. So It depends upon the complexity of your work, whether you are happy in just considering this kind of feature, or you want to take a mix of that. If you want to take a mix of that, that becomes slightly complex. So suppose you want to just take simple chemical properties of this molecule to build a machine learning model. And what we do in machine learning model is that we try to correlate the property of molecule with some of these structure. Like if a molecule has high solubility, what kind of atom composition it has, what kind of aromaticity it has, what kind of double bond and single bond representation it possesses. So we try to find out such kind of thing by applying, as I mentioned previously, it could be a simple clustering method. You cluster high solubility, low solubility molecule based on this feature, which could be like this. So you suppose in the bottom you have a molecule with low solubility. On the upper side, you have molecule with high solubility. And then you can say that, okay, a molecule with high solubility, the feature which is responsible is molecular weight. And then you say that, okay, so because I want highly soluble molecule, I'm going to put a filter that a molecular weight beyond this only I'm going to consider for my work. Okay. So such kind of decisions are made automatically by AI models. You don't have to do it manually. So you just give this table and it will give you which kind of molecule has certain properties and how it is correlated with its nature. Okay. So this problem could be simple regression, classification, and so on. Okay. And this ultimately have application in different kind of thing. It could be used for toxicity prediction and so on and so forth. So this is the overall framework, taking an input data, calculating some feature, and then applying some simple and complex machine learning model to make a decision. A decision could be a pure classification. Decision could be a clustering if the data is not labeled. A decision could be like a multi-class prediction. Suppose you are predicting, you have very complex data set where you have different classes of molecules. So each class will show certain properties. So instead of like, suppose you have 500 molecule, you don't want to test everything. You just take one from here, one from here. So each cluster you take one molecule and give to experimentalist. He will tell you what is the result, and based on that you decide that which cluster has a property which might be antibacterial in nature. So the bottom line is, rather than testing millions of molecules, by developing such model, you can test only few molecules and spend less money, and that could be very helpful for a large drug discovery project. Okay. So any question at this moment, something is not clear, please do ask at this point. If the process of applying AI is not clear, taking an input, calculating some feature, and then selecting which kind of model will be helpful in my case, and then getting the output. Any question or any doubts? Okay. If no question, then I'll go further. Maybe I'll skip this slide because I think this will be more complex. So if suppose we have built a very accurate model by using the previous process, then I will have this model in my computer, and then I will screen all the compound because I have learned what is the nature of antibiotic molecule, and this is I have saved as a ML model in my computer. Now somebody gives me a list of compound and asks, can you tell me which molecule out of this has antibiotic nature? I can quickly pass through my model and it will rank, or it will tell me a positive or negative value, which might tell that whether that molecule has antibiotic kind of nature or not. So these models can be used as a screening tool, and in the language of drug discovery, it's called virtual screening. So why virtual screening? Because generally this screening happens in the lab, but when you're doing this in computer, it's a virtual in nature. So you're screening compound not based on experiment, but based on AI and machine learning model. And if you have built your model very accurately, it is going to give you a very accurate result about the nature of your compound, whether it's antibiotic or not. Okay. So the bottom line is you can screen millions of compound in just 1 week. And then you have ranked results And then this can be go to the experimental people and they can do their task very quickly rather than screening this all millions of compound in the lab, spending time and money. So it has certain trade-offs also. If you have not built your model accurate, it could be have false negative and false positive. That we are not going to talk about these things here. But if you have not built your model very accurately, it can also give you wrong output. And or if you have very less data, like suppose you don't have enough data point to build a model, then the accuracy of that model also will be slightly questionable. And also many times, suppose you don't have access to a very large computational facility, then the conventional machine learning models require you to tell the important feature beforehand, like here. In this picture, I told you, right, you calculate from this drugs, this kind of feature, and then build the machine learning model. But suppose you don't know what features are important for a drug discovery or a novel kind of molecule discovery. Then the question comes, suppose I don't understand the molecule, this feature might not help. So in those cases, you have to apply a kind of model which does not require this feature calculation manually. So what I want is I just give my structure. A model should learn automatically which features to calculate and then give me the output. So this kind of model is what people are looking for. So the model which does not require manual feature calculation or manual input about which feature to calculate to build a machine learning model. I just want to give my molecule and the model itself decide which features to calculate and then build a model automatically. So this kind of models are called pure AI or deep learning based models. Okay. So this is what I'm talking about. For many times, just like you have seen the face recognition method, right? So on the airports, when you enter, there are face recognition method which can capture information about you, and automatically a check-in happens, right? So and also there are face recognition method. We can identify whether the picture is of a human or any other object. So how this method works, we don't tell them, right? So this is a human, this is a car. So how they work? So they work by something called they work based on a deep learning kind of architecture. So generally in deep learning architecture, we don't tell the model to calculate a set of features manually. What it does is it takes the whole input and try to calculate the feature which is giving the best output. Like this is a car. To predict this picture as a car, which feature is important? I don't have to tell that model that, okay, a car should have 4 wheels, 2 headlights, one steering, a roof, and 4 doors. I don't have to tell that. It will automatically it will automatically predict from the data itself which feature I should concentrate so that I can classify this object as a car. Or if there is a human, automatically which feature I should focus so that I can classify that particular person as a human. So automatic feature engineering is what people are currently using in drug discovery, that you give a molecule, it will automatically find out which feature is correlating with the activity, okay, or which feature is correlating with the binding affinity to certain target in a bacteria. So automated feature learning is what people are looking for. That for that you give a molecule, deep learning learns feature automatically and tells you whether this molecule is going to kill a bacteria or virus or not. So this kind of virtual screening methods are there now, like which is called you must have gone through in this evaluated course about docking, right? For docking, what you do is, so all of you know docking. I mean, you have gone through docking, right? So what you do in molecular docking?
Sir, we are yet to complete the course in that little bit we have lagged behind, sir. So
Okay,
they
okay.
could
see,
not.
see. Okay. No problem. Yeah. So I'll explain actually, like suppose let me draw here. Suppose this is a protein molecule, and then you have this triangle shape is a drug, right? So suppose this is a protein, and this is a drug molecule, and you try to find out whether there is an interaction between this drug and this protein or not by simulating the binding in a computer, right? Which is called docking, just like dockyard. So you have dockyard, right? Where the ship comes and docks, right? So something like that. So this is, suppose this is a port and this is a ship. So ships comes and dock at the port. But if the ship size is very different from the dockyard, it may not bind. Or just on other analogy is like this is your lock. Okay, so let me select the laser. Suppose this is your lock and this is the key. And you know very well that not all the keys fits into the lock, right? So a key and the lock should have a complementarity. So likewise, suppose this is a so suppose my lock is a protein which is important for survival of bacteria. I need to discover a key which is a small molecule which can kill the bacteria. If there is no competence binding between these two, suppose my lock and key are not binding very well, then this key is not going to work, right? Or in other ways, key is not going to kill my bacteria. So I need to find out whether this key is good for killing this bacteria, which is a protein inside a bacteria or not. So earlier, what people used to do that uh, we need to computationally dock this in all the location of my protein, and if it fits well, it has a complementarity with each other, then it have a good affinity. If it does not fit well, then it's a bad affinity, so it is not going to bind. So molecular docking is this process. You try to check where this small molecule can bind. So, and this can take a lot of time, right? And suppose we have, if we have, so the tool which you use is Autodoc Vina. So Autodoc Vina takes at least uh, 10 to 15 minutes if your protein is very large and your drug molecule is also large. So you can imagine that if you have to do this process for millions of molecules, it is still going to take a lot of time. So what deep learning can do that? In deep learning based screening, I'm not going to dock this molecule at all the location everywhere, right? I'm not going to do that because it's very time consuming. What I'm going to do that because I have a machine learning model, which is a magic. It has learned all the possible features for all the protein which exist in a database. I know that what could be the protein features where this molecule bind to it. So it has learned this from the existing data set. So then what I'll do that I'll calculate, so because this feature calculation is very quick, I can quickly calculate this feature about this molecule, calculate some feature about the binding interface. If the binding interface features is compatible with the small molecule feature, it is going to bind. If it is not compatible, then it's not going to bind. So without docking, you can check whether a molecule is going to bind to a protein or not using a deep learning based protocol. Okay, so there are deep learning methods without docking which can tell you whether a molecule is going to bind to a protein or not. And that protein could be a key protein in a bacteria or virus. So this is going to, and they don't ask me which feature I should calculate. You just give them the protein molecule and the drug molecule, and they can do the task by their own because they have learned everything about the protein structure and the small molecule. And they are well aware that if a small molecule need to bind to a protein, what kind of feature I need to look for. Something like this. Okay, so so this is one example where deep learning found or AI found its application. AI can also be used to generate new molecule. Like many times, I may not be just interested in screening molecule which exist in database. My requirement would be many times very novel, like you have AI generated picture which I shown in my first slide. Okay. Those pictures may not exist anywhere, right? AI have designed. So likewise, I might have very novel disease or novel problem for which there are not molecules which are already present in databases. So screening them might not help even I have very good machine learning model. So what actually I need is a model which can generate new molecules which have a very novel properties, what I'm looking for. Okay. Just like you had previously a models, like right now you have ChatGPT, you tell ChatGPT generate this kind of image, it can generate some not so perfect image, but some similar kind of image for you. So sometimes we want same kind of models for our drug discovery. So that I tell that I need a molecule which should bind to this kind of protein which present in bacteria, and the bacterial protein have this feature. So can you design a novel molecule for that? If this model has learned all the chemistry of drugs, then it should be able to ideally do that. And that is the one target for other kind of AI molecule, which is for generating new kind of drug molecule rather than screening them. Okay. So these are the different kind of problem I'm telling you, like screening already existing drug using AI method and designing new drug from the scratch. So those are the two different kind of problems. So you might be interested in screening what is already existing. You might be interested in generating totally new molecule based on the problem you have in hand. Okay, it depends upon the problem for which kind of AI methodology you want to use. And generally, these methods, I'm not going into detail of these methods works on the principle of something called graph neural networks. Okay, and just like you have drawn graphs in some point of your time, so you can draw graph if you have certain data. Likewise, you can draw a graph from the structure of a molecule also. Okay. This graph will have information about how this particular atom is connected to this. I can generate a graph using this also, right? Suppose I show you example like, so this is a graph, right? And this is what it is. It is what kind of pattern it is capturing. It is capturing pattern of whether this atom is joined to this atom or not, or this atom is connected to this atom by at least 2 bonds. Likewise, so this is a graph representation of molecules. But generally, the AI method which try to generate new molecule, they encode the structure of this complex molecule in the kind of graph, and they try to build these kind of models, which is called deep learning model, based on the graph representation of molecule. Because, and the question comes, why we need to represent this as a graph? Why not a structured cell? Because learning on a graph is easy compared to learning on the raw structure. Like, so because graphs have just connectivity, and based on the connectivity, you can define a lot of important properties. And also you can define a patterns in graph. Like, so suppose you have pattern which is 2 atoms and patterns such as 3 atoms. It is easy to do on a graph rather than the raw structure. So many of these techniques of molecular property prediction or new molecule generation works by converting the original these molecule into a graph representation and then try to do this process. So now I'll show you one example, like how people have used this kind of model for antibiotic discovery and why it is required. So given the problem of AMR, I mean, we all know that, and I have already discussed in this a couple of minutes back that we are facing a problem of these antibiotic which is shown in this picture. These are used for decades. But many of these antibiotics, like carbapenems, if you talk to any physician, they will tell you that they must have treated some patient which are not responding to this even very advanced antibiotic like carbapenems, or even a very advanced antibiotics called lipolipids. So slowly, due to different kind of reasons, so bacteria are developing resistance. And these resistance are causing this antibiotic not to work. And if these antibiotics are not working on a patient, we have very less option available. And there are a lot of deaths which is going to occur in future due to this kind of resistance. So we need new antibiotic to be discovered at very fast pace. But many pharma companies have very less interest in discovering antibiotic because the process is obviously lengthy, and they have to spend money, and antibiotic cost is not so high. So they're not going to earn a lot of money, unlike other kind of problems like cancer, genetic disorder, or immune-related disorder. Those are the diseases of wealthy, and what is considered that the infections are not so economically relevant area for many pharma companies. So there is a very less focus on discovering new antibiotic. But whatever repository of antibiotic we have currently, many of them are not going to work after 5 or 10 years in many of the patient if they have a problem related to resistance. So how we are going to tackle the problem of resistance and how people are doing some work in speeding up the discovering of new antibiotics so that people may not die in future even with simple infection by simple bacteria. So and these antibiotics are used in a lot of quantity in India, right? People also take antibiotic without prescription. So antibiotic resistance has that is totally different kind of topic. It have multiple reasons, right? So it could be uh, due to unregulated use of antibiotic, but also antibiotic is reaching to us in different other ways through diet, through dairy, through environment, and so on. So there are multiple factors which is causing bacteria to develop resistance. And this resistance is going to be very lethal in future because we have very fixed a basket of antibiotic, and many of these baskets uh, are still show very less efficacy on bacteria which have antibiotic resistant genes. So how are we going to tackle antibiotic resistance? The only way is to discover new antibiotic very quickly, and it can be only be done quickly using the application of ML and AI, because if you're going to the conventional route of antibiotic discovery, it is going to again take 10 or 15 years, and that much time we may not have to wait. So how we can make antibiotics faster? This is one case study from Broad Institute. What they have done is they have taken different kind of drugs, uh, not only antibiotic, but all the approved drugs by FDA, and try to find out whether there is any documented proof about their antibacterial activity. And they have also taken compounds which are in experimental phases. So they have taken data from about 2560 molecule, 2560 molecule, which were showing certain antibiotic kind of properties, and they have selected a very simple bacteria, E. coli. So what they have done is they had some data related to these 2560 molecule, and they try to learn what kind of feature this molecule possesses so that they are able to inhibit the growth of E. coli. So what they have done, this is a simple kind of table, like you in one column, you put all these 2560 molecule name, and then you in other column, because you have experimental data about this 2560 molecule through lab, you code that whether it's antibacterial or not by some assay which you have done by them on E. coli. Then you can say yes or no for all these 2560 molecule. Then because this molecule must have some structure, you can calculate, you can just fetch the structure from any database, and then you try to build a model to correlate how these drug molecules are showing antibacterial nature. Which feature? I'm not telling this model which feature to calculate. It is going to correlate these two things. How antibacterial nature, whether yes or no, is correlated with this drug. So if I can learn what features of this drug makes this antibiotic and which feature a drug is not making them as antibiotic, if I can learn something about that, I can use this model for classifying molecule which might have antibiotic properties, right? So I have just spent very small money on testing these 25-60 molecule. But now, once I build this model, I can use this model to screen, suppose 1 million compound. And from this 1 million compound, I can give that, okay, these are the top 100 molecule. You should go for testing. So it's a great kind of work which was done by them. So what they have done, using just 2000 molecule, if I can build a simple machine learning model which can correlate with the nature of the drug and its antibacterial nature, then I can use this model to screen a large library of compound and maybe take top 100, top 200 compound to test them experimentally. That is going to save a lot of time and money. So traditional approach was like generally using handcrafted feature, but they have used not traditional approach. I've already mentioned they have used approach where it does not require you to tell model which feature to take. It will automatically learn the feature and tell you the final things. So this kind of approach is what we need. So but that required the collaboration of experimentalist and computational biologist. Okay, and if you are a student, I mean, these papers I can send you if you are further interested to look into detail how to develop this model, and but it requires some basic understanding of computational chemistry. Or even if you're not interested in chemistry, you have you know simple medicinal chemistry of drug molecule, then you will to do that. Okay. And then simple, you don't have to learn programming from the scratch for doing that. There are models. You can use a already existing model. You can have a novel kind of data. Suppose they have discovered against certain class of suppose gram-positive bacteria, but you might be interested in doing something on gram-negative bacteria. So then you take suppose molecule which have shown some effect on gram-negative bacteria, try to develop that model, and then try to see that whether that model can be used for screening similar kind of compound from a database. Okay, so that's what a good solution could be. I'm not going to this, but I'll go through. So a lot of other research is going on, which I'm going to skip. I'm going to quickly now. So to my other part of the talk. So this is what ultimate the bottom line is. You have two approaches which is possible, which can learn feature automatically, and or you can have a possibility of handcrafted feature if you know that which feature is important for a certain activity. So based on what is available, if you have a novel kind of molecule library where you don't know what feature is important for its activity, you may want to use a model which does not require a feature to be given as an input. If you have a feature already available, like if you have antibiotic molecule and you want to train your model on antibiotics which are already approved, want to learn which feature in antibiotics is important for its efficacy, then you can have a model which required a feature to be calculated and then then given as an input to a model to learn the properties of antibiotic and its efficacy. Okay, so a lot of this, I've shared this slide, but all this work was not theoretical. They have tested all this molecule and they have shown that. So the approach which I discussed in the previous slide, they have tested experimentally and they found that the model works very well and they could discover this molecule which is called helicin by screening a large database which is showing very good inhibition of E. coli in vitro experiment. So things that it's not theoretical or bookish knowledge. These model works in the lab. So, and that is shown by this assay, which is called optical density. It will show you whether how many cells, I mean, if the cells are alive, they have different kinds of optical density, very high optical density. If the bacterial cells are dead, after the exposure of any molecule, the optical density will be low. And that's what it's shown by here. The inhibition of the bacteria if in vitro it is exposed to this molecule, which are discovered by the deep learning model, which I've shown in the previous slide. So these model works in the lab, and that is why it's very useful. Even suppose you have experimental collaborator, if they want to screen certain molecule without spending a lot of money, but these model can be developed for different kind of test cases. Okay. And they have also tested that in mice system, and it was also shown very good result, not only in vitro experiment, but it's also in the mice. And they have exposed this, infected mice in this particular discovered small molecule, and it has shown that the infection was gone after a certain period of time. Okay. That shows that the efficacy of this molecule, it was very good against a certain kind of bacteria, but other kind of bacteria it may not be. So you have to be very specific in discovering this molecule. You cannot discover a magic bullet which is going to work on all bacteria. Like in this case, it was very well working for Acetobacter baumannii, but it was not very well working for other bacteria. Okay, so bacteria is still not. So the thing is, these drugs are very specific also. Okay, so what they have discovered is these drugs are very specific against one bacteria, but they may not work against other bacteria. What they try to show. So this tells you a lot of positive message that how people have discovered very effective molecules using the machine learning or AI based application, and that's what people want. So the second part of my talk is like, right now we are just mostly concentrating on the molecule itself, right? You have a problem, but there are models which take into account about the targets, right? So, and both need to be feeded into AI model. If you are developing good AI model, then you're going to feed the information about the drug and information about the target both at a once, if you need to discover how this is going to bind to this. But the problem sometimes is that this structure might not be solved for a particular bacteria or particular virus. So in that case, there is no way. Earlier, there was no way to consider this combination for discovering a drug. Many times people just used to screen. Suppose I have antibiotic, I just screen similar kind of antibiotic and then test whether it works or not. Because the target structure many times is not available. But now we have methods through which you can determine the structure very accurately. Henceforth, you can give both the chemical molecule information and the target information in the same system, and it can discover a better molecule, because we know that most of these small molecules are going to target a protein or a receptor. So it's good to have the structural information about a target also into the model, so that your accuracy of what drug you're discovering is high. And that is what my other focus is, how people have discovered very accurate kind of model to discover, to design a protein from the scratch, or try to predict their structure from the scratch using AI. So this I'm just skipping for the time because I have less time. I'm going to quickly. These are just extension of that. I'm going to show you the second part of my work, which is my presentation, which is about AI for protein design. And yeah, before going into that part, I'm just going to show you what is the current status of AI design drug in world. So these are the drugs which are currently in different phases of clinical trial. So the phases are mentioned here, and a different kind of disease they are targeting. So if you are really interested in this area, so what you can do is you can pick up, because all these drugs already, some part of their discovery is already published in multiple papers. So if you are really interested in this area, what you can do is you can pick up any drug molecule and try to see that how these organizations have discovered these molecules starting from the scratch and how they have reached to phase 2 to phase 3 trials. So each of these drugs, we have associated papers. And if you're really interested in this area, what you can do is just pick up that paper and try to go through that, understand how they have discovered all these molecules against different kind of disease condition. Okay, so many of these drugs are in the late phases of clinical trial like this. So coming to the second part of my talk is AI in protein design. So right now I just focus on drugs, small molecules, how this can be used for discovering novel therapeutics. But other area which is very important right now is AI for protein design. And protein, you know, right? What protein is? If you are from biology background, then you must have undergone this kind of information in some of your courses. So you have DNA, then you have mRNA, and then a protein is produced. And protein is made up of 20 different amino acids. And when the protein are produced in the cells, they are in linear form. But for their function, they need to fold into this kind of 3D complex shape. And this shape is key to their function. If the protein is not folded well, it cannot perform its function. Okay. So nature knows how to fold a protein, right? But as a biologist or researcher, if I give you a problem that if you have a straight, this unfolded amino acid string, how are you going to fold into this 3D architecture? Any response from the student? Like suppose I give you this 20 amino acid strings of protein, how are you going to fold into this 3D structure? Because we know in the biology that without folding, no protein can perform its function. The example is hemoglobin, which present in your blood. So hemoglobin cannot work if it is not folded into its right conformation because it requires heme binding, and heme cannot bind if the protein has not adopted a right 3D structure. And you know that there are multiple diseases which occurs when the protein has not folded into its right conformation, or a protein get misfolded due to some genetic mutation or due to some other reason. So if protein misfolding occurs, a lot of problems occurs in our body. The question is, given a simple string of amino acid, how this amino acids or different combination of amino acid knows that it is going to fold into this 3D structure? Anyone from the students? How nature knows that this?
Please respond, students. Raise your hands or unmute yourself and speak. All the participants of the value-added course can speak. I think you have completed on these topics. They are typing as chaperones, sir.
Okay. Okay, chaperones. Okay, very good. The chaperones are there. Okay. All the protein required chaperones for folding.
Please respond. Raise your hand and respond.
You can speak. Don't feel shy, or you're sleepy. Then it's
Not like that.
because I'm speaking. I don't know what is going on other end. So all
Yes.
mute and all camera is off. So we cannot see what they are doing.
Yes, sir. Yes, sir.
That
Yeah.
is difficult
Students,
thing in.
all the participants who are there here, a value-added course or otherwise, please respond to the query.
Okay.
They have written a specific amino acid sequence.
They have typed in chat. Let me.
Yeah, they have typed in the chat box. Sir.
Okay, let me see what they have typed. Specific amino acid intermolecular interaction. Okay. Yeah. So we have 55 students, but only one answer. Okay.
3 have
Yeah.
responded.
Yeah. So I hope. Okay, that is a good hint. Okay. So given a string of amino acid, this protein folds by the nature of some interaction, right? I mean, all this amino acid can be classified into certain group, right? Some are hydrophobic, some are hydrophilic, some are charged, some are not charged, and even charged, you have positive and negatively charged amino acid. So there are biochemical rules for folding, right? But and that is true. I mean, you have positively, negatively charged amino acid, they will obviously attract to each other. And if they are amino acid of same charge, they will repel to each other. And then there will be hydrophobic amino acid that will form the core of the protein. So there are underlying biochemical principles which can decide how protein is going to fold into. But given the diversity of the sequence, we have millions of protein sequence, but the protein folds which we see is not in millions. They are very unique fold and much less compared to the number of sequences we have. So the question comes, how different sequences might be folding into same kind of architecture? That is sometimes very difficult to predict. You have 10 different protein sequences, it might fold into very similar kind of 3D structure. So that is a very challenging problem. Like obviously there are physicochemical rules to decide the folding, but if the protein is super large, suppose you have 1200 amino acid, then it's very difficult to fold that 1200 amino acid into a compact structure. So the problem is there are physicochemical principles, but those physicochemical principles not enough to explain the folding pattern which we see in the nature. Okay. And there are protein which also sometimes folds when some important function they have to perform. There are classes of protein which are always unfolded. So there are some unsolved mystery that how protein is folding. People are trying to solve from 50, 60 years. And a very complex problem. People are working from number of years, lots of paper came, but there no clear distinct. So there are theories which tells that hydrogen bonds are important. There are theories which which tells that hydrophobic interaction are the governing interaction. A lot of debate was happening. So how AI have addressed this problem, I'm trying to explain you in couple of minutes. And this why Nobel Prize is given to this kind of discovery because of the importance of this folding for different functional point of view. Like this folding might be important in binding and recognition, which is a very fundamental biological process, whether it's infection, whether it's an important biological function in your body, whether it's for transport or any other kind of function, catalysis. So a lot of chemical reaction goes on in every kind of life forms. And many times this catalysis happens in some binding pocket. And those binding pocket can only be formed when the protein is in folded nature. Mechanical function. You have collagen in your skin, you have keratin in your hair and nails. A lot of mechanical function can only be performed by this protein when they are in folded form. So all this molecular function can only be explained if you understand the structure. Just by looking at the sequence, sometimes very difficult to tell how this sequence are performing a molecular function. So solving a structure from a sequence is very fundamental problem, very important problem, and therefore people have received Nobel Prize, multiple Nobel Prize on solving this problem. And now people have gone one step beyond, not only folding a natural protein sequence, but designing protein from the scratch. Like suppose I want a protein which should bind to a receptor on a cancer cell. That protein may not be produced by nature by itself. I only know that I need a protein with this kind of function. Can I design this protein from the scratch? That's the extent people are now into. Now it's possible to design this protein from scratch also. You tell me what kind of protein you want to bind to, I can design that protein for you. Okay, just like a tailor. Tailor can take the measure and can stitch the cloth for you. Likewise, I just need the measure about the target. I can design a protein which can bind to that target. And that protein, if not hypothetical, you can produce that protein in the lab also. That's the extent people are working now into this field. So given this context, and given the huge diversity of protein, it's a very fundamental problem to solve the protein structure and also design this protein from the scratch. And if we can do that, we can revolutionize the whole medicine, whole industry to a great extent. Because what people want is good enzyme, good catalyst, good medicine, which can work and target based on our requirement. Okay, so this slide, I'm just showing the diversity of protein. If you have not done a structural biology bioinformatics course, there are multiple courses on NPTEL, Coursera, which you can take and familiarize yourself with this kind of structures, what they mean, and what complexity they contain. Okay, and this will be very useful if you're interested in this area of drug discovery. But drug discovery is not only about learning about the chemical molecule, it is also about learning about the target, which is often comes into this kind of diversity. It could be antibody, it could be a structural protein, it could be a membrane-bound receptors, or it could be a long protein and the fibrous protein like collagen. So there's huge diversity of protein, and this protein word is very interesting. And if you're really interested, I'll suggest you to take some good NPTEL, Coursera course, or do some project related to the protein structure. Then you can become more comfortable in this area. So stages of protein prediction. This is the story 10 years back. People used to predict the structure by looking at the homology. How it works is that I have a protein sequence. I will find out which protein sequence it is similar to. I'll give that information to computer. Computer can predict the structure for me. The problem becomes complex if I have a protein sequence, but there is no homologous protein present, or the homology is very weak. What I mean by homology is like, suppose you align these two sequences. If the similarity between the sequences is less than 40 to 50%, this protein cannot be called homologous. And if you try to predict the structure of your query protein based on bad homologous protein, then the structure is also going to be wrong. So people who are facing a lot of problem, like if you have a novel protein in hand, how are you going to predict the structure was a challenge. Okay, the only approach was available 10 years back or 5 years back was you discover a homologous protein, and if people have understood that protein very well, you try to solve the structure of your novel protein by learning from the structure of that. And that is the conventional procedure people used to follow. But now, so now what people have done is we had something called protein data bank, which has close to millions, close to millions of experimental structures solved that contain a lot of important information about the structure. So what AI people have done that they have used all this knowledge which is available. So protein data bank have very rich knowledge of highly diverse protein structures. The question comes, if I have a good computational facility, can I learn how this structure was folded given their sequence? So earlier we discovered that given a molecule, how it can bind to a target. Here, the problem is given a sequence, what kind of structure it can fold into. We had these resources already available. So what they have done that use these resources to create something which is called alpha protein structure database. So this is the database which have experimental structure solved. And the AlphaFold is a database which has all the protein structure available for which the sequence exists. Okay, so this is comes into the total number is around billions of structures. So you have only millions of structure there, but now you have access to all the protein structure which exists for which the sequence exists. It could be mammalian or it could be bacterial protein. So this has revolutionized the field, and that is why these people who were the discoverer of AlphaFold got the Nobel Prize. So earlier people used to only study the structure what is there in the PDB, but now you have in just one click you can download all the protein structure which exist on this planet. Okay. So the level of complexity they tried to solve was huge, and I'll try to just show you that what they have done. Okay, and also some related studies. And these are the other studies which have done something similar. Swiss model was again homology based repository, which people are maintaining since decades. This contains homology based protein models which are solved. So these are the databases which can be very useful if you want to apply machine learning or AI method to work on how a protein can be folded into a 3D structure. I will try to, and this is the slide which explains what they did. I'll not discuss all the complexity of that, but I'll try to explain you into very simple way what AlphaFold does. Again, I'm emphasizing AlphaFold is a database which contains all the structure of protein which exists on Earth. Okay, and that is from bacteria to mammal. Earlier, it was not possible to solve a structure to this extent. It was a humongous task. So what strategy they applied, how they can solve all the protein structure which people were suffering to solve since years. They have combined very, very smart strategy. So this strategy was twofold. This is what is mentioned as genetic database search and structure database search. So through genetic database search, suppose we have a sequence in hand, I'll try to find out whether this sequence is homologous to any given sequence in this database or not. Okay, I can make a search and I can rank this search where suppose there's a human protein. Human protein can match to a human protein or other mammalian protein or a bacterial protein. I will get this matching information from this database. I will also match this sequence to the structure database like PDB, and I get all the information that this sequence, how it matches to the existing sequence for which the structure has already been solved. And somehow I'm going to combine these two information. And this combination is what it paid for. And this combination is not random. What they actually see that what useful information I can take from this database search, which give me the information about the structure, and what information I can take from this homology similarity. So if I can combine the homology similarity of my sequence and the structure similarity of my sequence, if I combine these two, it might lead to a structure which is very close to the usual structure. So these two strategies they try to combine, and this was done iteratively. If they are getting something bad, they try to refine again by doing a better homology search and better structural search. By combining the strategy multiple times, what they found that they could get a structure which is very close to the experimentally solved structure. Okay, and going into more detail because we do not have background, I don't want to discuss this, but this technique of combining this in a contextual manner was done by a deep learning model which is called transformers. Okay. So if you are really interested in how AlphaFold works, there are documentaries on YouTube, lectures on YouTube, which can give more detail about how it is possible to combine homology and the structural similarity by using efficient deep learning based method. That is the only innovation they have done. They have efficiently combined the homology and structural similarity to get the final 3D structure, which people are trying to do independently. People either used to do homology alone or structural similarity alone because there is no straightforward way to combine these two different kind of information because structure is totally different from a homology, which you must have done multiple sequence alignment. If I give you multiple sequence alignment and ask you to combine with a structure, it's not an easy problem, right? That's what they try to solve. How multiple sequence alignment information can be combined with the structural information. If you could do that, you can solve this challenging problem very efficiently. So this was a success. They could efficiently combine given the deep learning advancement, and this is the same model which ChatGPT uses. Transformer model is what the model which ChatGPT and DeepSeek they are using. So this model, the same model which people are now using in ChatGPT, and you know that ChatGPT works by finding the contextual information. You type something, it will find out the context of what you are asking and try to give you some contextually relevant information as output. The only challenge is to find out how we are going to build a context between the multiple sequence alignment and the structure. And that can be done by this model, and that's what they have done. In a simple language, that's how I can explain this thing. And likewise, there are multiple other models which came into picture like ESM2 and this thing. So now the bottom line is we have a lot of such deep learning based method which can solve the problem of predicting all the kind of protein sequences in very quick manner. And if you want to use this model, it is, I mean, AlphaFold have maintained a database, but these two other models, they are available as an open source code, which can be run by anyone on any computational platform. So the problem of solving structure were more or less solved by them, but there are certain other limitations which I'm not going into detail, that many of the structure which are predicted by them may not be of good accuracy. Okay, that we can talk about later. So this is the interface of AlphaFold server. You can give a protein, suppose we have protein sequence in hand, you just paste the sequence in this window, and you can predict the structure in 5 to 10 seconds. Okay. And that protein could be anything, a very novel protein which nobody has solved a structure for. And this AlphaFold server also can predict a complex structure like protein in complex with DNA, protein in complex with RNA, and protein in complex with ligand. Okay. If you want to predict a complex structure like a protein bound with another molecule, that kind of complex structure can be predicted without doing docking. So that is the level of advancement we have made in protein structure solving and design. Okay. And this is the URL of this server. So you can go to AlphaFold server, just paste the sequence. And if you have suppose a DNA sequence which bounds to protein, suppose a DNA protein complex, and that structure can be downloaded easily from this server. Okay, so a lot of problems can be solved quickly, and you don't have to do any docking for many of these tasks. And many times the results are very, very accurate if you don't have a super complex problem. Okay. So the problem of predicting a structure more or less solved. Only problem comes with if you have a very complex protein in hand, many times the solution is not so accurate. Coming to another aspect that which I'm discussing, that solving individual protein was now more or less solved. Now people are focusing on designing new protein given a target, right? So this is the protein which people are working right now. So if you have protein, can you design a protein which can bind to it too? Okay. And can you design many of this? Or can you design multiple protein which can bind to the same protein? Okay. So this kind of problem is called protein design. So given a target protein, can I design a protein molecule? Can I design a peptide molecule? Can I design a small molecule which can bind to that without looking at any database or any sequence? Okay, that is the challenge people are right now trying to solve. And this is important from different therapeutic point of view, which so this is one example people have worked where so cancer is a challenging problem, and many times this is the picture of tumor cell, and on the tumor cell we have different kind of receptor which are expressed. Okay, and many times these receptors are very specific to bind to other kind of receptor. Like in this case, this tumor cell have PD-L1 receptor, and that bind to PD-1 receptor on these T cells. And that inhibits the tumor cell killing multiple times, because once it is bound to this tumor cell, these T cells are no longer available to attack tumor cells. So what people have done is, can we design a protein which inhibits the T cell binding to this receptor, and therefore these T cells are available for killing multiple other tumor cells. So in that way, we need to design a protein which should bind to some of these receptors so that this is not available to bind to PD-L1 because it was inhibiting the tumor killing. And because this is not binding, now this T cell can kill the tumor cell effectively. So here what we need to do is we just know about the T cell receptor protein. And I don't have any information that because nobody have discovered what kind of protein can bind it to. So I'll just give the information about this receptor, and there are methods which can design a novel protein which can bind to this PD-1 receptor on the T cell. And this could be a potential cancer immunotherapy strategy for different kinds of cancer. Okay.
Sir,
So
there is a
this,
question in the chat box, sir.
yeah. Yeah, hello, ma'am, have you missed anything?
Yeah, can we make a drug using this AI/ML technique that can selectively kill cancer cells while leaving the other healthy cells intact?
Yeah, it depends upon the context. So right now, suppose, I mean, we are working on a problem where we are trying to, it depends upon how much you know about your target, right? So like here, suppose I would just want to target a cancer cell. So I need all the information about what kind of receptors are expressed on cancer cell, right? So healthy cell have different kind of receptor expressed. So what I mean by receptor, these kind of molecule. If I know what kind of specific receptors are expressed on cancer cell, and if these receptors are not expressed on cancer cell, then we have a strategy to do that. If you do not know which specific receptor are only expressed on cancer cell, in that case, it's difficult. You target your cancer cell and then it is going to also kill some healthy cell. And that's what happened in most of the chemotherapy drugs, right? So you must have heard about cancer patient, right? So chemotherapy is the only option in the later stage of cancer. And many of these chemotherapy have a lot of side effects. Okay? And these side effects happen because these chemotherapy do not only affect the cancer cell, but also a lot of healthy cells. They're not very specific in many cases. So if we want a very robust chemotherapy, which should not target a healthy cell, what we require all the molecular information about the receptor which are expressed on the surface. I have to take the biochemistry of those receptors into account while designing a molecule which should only target the receptor which are expressed on the cancer cell and not on the healthy cell. Okay, so that biochemistry need to be learned by AI, and that you can learn if you have a lot of receptor information. What I can do, I go to a database, find out what all the cancer receptors are expressed on, suppose lung cancer. I will extract all those receptor information, try to learn their structure, sequence, and then try to design a protein inhibitor so that there is only very high specificity between the design protein and the receptor expressed on the cancer and not on the normal cell. Okay. So this required a lot of effort and also a lot of understanding about the receptor biology, receptor biochemistry, and the cancer biology also. Okay. Without taking that into account, if you just randomly apply any AIM, that is going to fail. Okay, so this is what people are looking for: novel protein design as a therapeutics for different kind of diseases. This is cancer is just one example which I'm showing. Okay. So how this works? This works on the place. So what I try to explain is the strategy. So this works. Novel protein design works on the strategy of this picture which I'm showing. You must have played different kinds of such games, right? Where you have a picture and you have to disintegrate all the parts of the picture and then you can reassemble based on certain intuition, right? So deep learning also works in the same principle. What they do is they try to, so we have this original picture. We'll keep adding noise in this picture so that it becomes indistinguishable from the original picture. But while adding this noise, I will learn what kind of noise I'm adding into. It's just like you have a complete puzzle and you disintegrate all the puzzle, but you have certain feature by through you can reassemble them. If you learn this process that assembling a complete picture and disassembling that multiple times for that you have become expert, so it will be very easy for you that anybody gives you any picture, you can quickly disassemble and assemble. So this kind of biology, this deep learning model try to learn. They try to learn uh, like you have a receptor protein available. You have all the biochemistry of that protein available. So they try to learn from the scratch. I have this target, I have this protein molecule, and all this information I have present in the form of picture. I will try to learn how this protein molecule can be designed from the scratch. Right? Suppose I have this particular receptor, this is a protein. I'll try to add more noise so that it becomes indistinguishable that it can identify my target, right? So I keep on adding noise so that it can no longer bind. While doing that, I'll keep on noting that what I'm doing, what I'm doing on this part of my puzzle. So if I learn this process by adding noise, ultimately I'm going to learn how to design something from the scratch, right? It's just like you become so perfect in reassembling all this puzzle so that anybody can wake you up even from your sleep and you can reassemble this picture quickly. So this kind of machine learning models are there, which works by adding noise to the original pattern. And by doing that, they try to learn how a pattern can be made from the scratch. Okay. I hope you understand the analogy like from this picture. Consider this picture as a binding partner to a receptor. A perfect picture is a perfect binding partner. And the not so clear picture is a stage where this binding partner become a non-binding partner. So from this to this pathway, I'll try to learn which features I have to accumulate to make this protein as a binding partner. If I learn this pathway of going from the perfect picture to so bad picture multiple times, anybody can give me any target. I can design that protein from the scratch. So that is what in simple language I can explain to. So this is called a diffusion model. So you try to add something called Gaussian noise while designing a protein. Suppose this is your target. So you keep on adding noise so that a target, your molecular protein no longer binds to the target. While doing that, you keep on noting that what you're doing. So while doing this back and forth process, if I repeat this process hundreds of times, I may learn that what kind of process a protein, what kind of sequence of protein adopts so that it become binder to non-binder. So if I do this millions of time on all the structured information available, I can become an expert. I know that what sequence I need make to protein bind and non-bind. So if this kind of learning can be done by any AI model, so it will become very easy for you that you give a protein, it will quickly find out what kind of sequence I need to design so that it binds to this protein. Okay, so something like that kind of conditioning people do while doing training this model. So this model called RF diffusion model, that is the prize this year David Baker got for designing this kind of method for protein design, for designing protein from the scratch. Okay, so you can only design something from the scratch when you understand how the real protein looks like. And to understand how the protein looks like, you need to be mastered that if I give you disassembled protein or amino acid, how you can reassemble back to the 3D structure. If you don't know how to do that, you cannot do that. So these deep learning model understand if you give me disassembled amino acid sequence, how you can combine to fold into a right kind of 3D structure. Okay. So this is again a lot of, so these are all the computer designed protein inhibitors against PDL1, which is a very important cancer receptor. And all these were tested in the lab. And you have multiple protein candidate which are designed by AI. and those works very well in the lab with different affinity. So some have very good affinity, some have slightly not so good affinity. So in this way, lot of effort can be saved, right? You don't have to screen through multiple millions of protein molecule which exist in database to find out which protein is going to bind to my receptor. I'll just give me the final receptor, and this kind of model can find out, oh, this protein is going to bind well, because I have learned that what it required to bind to certain protein to this kind of receptor. Okay, so this kind of strategy people are using, that you give me the target, I can design which protein it can bind to. And then experimental validation shows that this strategy works in the lab also. It's not theoretical. Okay. Okay, so this is what all I wanted to present quickly. I mean, so these are the final picks, and then they have compared so the structure binder from for which the structure is available, and it matches very well with the design protein inhibitor for PDL1. That shows these AI methods are not theoretical, or they work only in theory. They works in principle, and this all experimental validation is the proof of that. So experimentalists are now slowly adapting to accept AI and machine learning designed proteins and inhibitor to test in the lab. Okay, and that can save a lot of money and effort, like in this case. Okay, so there is a lot of other things which I'm not going to talk about. So life sciences, there are a lot of other applications for machine learning and AI, and you might be interested in totally different domain, but this is a good area to work with, but it requires certain sincerity from you. I mean, if you're really interested in this area, you have to make a good effort in learning some basic courses through, and there is no dearth of resources online. I mean, you don't have to even pay for that. Lot of online courses are there, online tutorials are there, which you can follow in the guidance of someone to complete this task if you're really interested in research in this area. Okay, so these are the other areas where deep learning and AI methods people are working related to antibody, nucleic acid therapeutics, cell-based imaging, vaccines, and so on. So those I'm not going into detail of. Okay. So I'll quickly show you. So these are the, practical resources. So these are the GitHub link, which I'm copied here. So this provides you some tutorials in the form of notebooks, which you can execute on your laptops. It just required an internet connection, and you can just follow some tutorial online. Just search on the tutorial how to run Google Colab notebook on internet, and then it will give you the whole guidance how to use these notebooks and how to do this thing on your laptop itself. Okay, if you don't have to purchase any software or wealthy computer for that. So these are the practical approaches. So some of these tutorials are available at this link, and if you want to try this protein design or some of the already existing project, you can follow this link and try on your laptop itself. Okay, so I'm not going into much about, so these are some of the work from my lab. So we have developed a server, ML-based server for screening peptides against anti-hepatitis C virus. And this came recently in American Chemical Society journal. So this server is openly available, and this paper also, if you want to read how we developed this model, I can share with you. So these are the work from my own research group. We have developed this machine learning based server to screen peptides for anti-hepatitis C. We are also working on snake venom toxin. We have recently designed certain peptide inhibitor for targeting snake venom toxins, because snake bite is a very huge problem in India. So we have working on designing novel peptide inhibitor which can target different enzymes in snakes. And it's a very complex and challenging problem. So we are also working on this point of view. And we have proved that, and we have experimental computational result to show that the efficacy of what we have designed is very good. So if you are interested in more detail about the work, you can contact me offline. I will give you more study material, and if you want to go into in depth of any of these projects. We are also working on discovering novel antibiotics against bacteria. So we have certain peptide candidate which we have designed, which showed very good affinity against many of the very important bacteria which is causing AMR problem. So this is third work which we are doing in our lab. So these are the focus of our lab, designing novel peptides which can be against virus, snake venom toxin, and bacteria. So these are three works which currently we are following in our lab. This is our group. So most of the work performed by Ayushi, Rashi. So these are our PhD student. And these were two student trainee which visited in summer, and a lot of this peptide discovery work have been performed by them. And this is the Center of Life Sciences, and we have a PhD program which is funded by university, and likewise we have MTech program and MSc program. So MTech program is also funded by a fellowship if you have qualified any national level exam or you have good CV. So, and yes, if you have any question, because this is 2 hours, is very small time to cover these things depth. I hope I have made justice to that. And if you have any question, you can write back to me, and I'm happy to reply. Okay, that's it. Thank you.
Thank you, sir. I think you have condensed a lot of information and in a very simple and subtle way where basic knowledge people like us would like to understand or would be able to uh, go into it. I think all the participants here, I'm sure they would have definitely benefited. Any
Yeah, I mean,
questions?
it's a complex topic, but I try to give you a flavor. I mean, you don't try to understand everything which I delivered, but even if you can take 10% and work on these things, it may be helpful in your masters or long term, right? I'm not expecting in 2 hours you learn everything what I talk, but even if you can capture 10% of the thing and try to do some self study or try to take project in those direction, it can help you in long term. Yeah.
Any questions from the participants? Quick questions for sir. Any questions, students? Please unmute yourself and ask. I think they are.
Yeah. They might be waiting for, okay. There will be any potential cybersecurity risk associated with using AI in drug discovery in long term. Okay. So I think what you're asking, like, suppose, okay, so if I'm using some proprietary information which I don't want to share with the service provider, or could it be a challenge? Yes, and therefore, like what we generally do, I mean, when you have a proprietary information or the information which you don't want to share with the external service provider, then you can do it locally. I mean, a lot of programs or a lot of other software, if does not require internet access many times, you can locally install in your computer and run it. But obviously, if you want to run, if you want to do a serious work, then you should have at least some access to high performance computing, like HPC servers and these things. Okay. So yeah, so high performance computing, if you have in your institute itself, or you can get from some other academic institution, then the risk of this thing can be minimized. Yeah, if you don't have to share your information publicly when you're running program on a server. But most of these servers are made by academic people, and academic people don't have any such interest that to capture your data or to use your data for some other purpose. If you're using some software which are provided by some other organization which may have potential interest in what you're doing, then it can be an issue. But yeah, cybersecurity risk is minimal if you're doing on your local laptop, local system, even if you're using outside server, the risk of, unless and until you're exposing your data to outside, there is no such risk. Okay. And if you are very private information, or you don't want to share any data, then it's good to have your own server. That is what we can do.
Participants can respond if they have how far they have understood the lecture and how they have felt about the talk.
Sir, I would like to ask a question. You're talking about docking and comparing it with deep learning, where you're able to dock a protein with a ligand and get the output by using deep learning mechanism or the models. When we are doing docking, we are able to predict the interaction between them by finalizing the energy output, how we are getting it and how they are able to dock with each other. That's output based on which we are able to predict which ligand is the best one. But in deep learning, how do you predict that this is the best one, which you can select that is a ligand having a good interaction with the protein? So how do you predict and get the output for that one, sir?
Okay. So I mean, suppose I give an example, like, so Autodoc Vina we use. So Autodoc Vina is, I mean, if you're using docking, so Autodoc Vina give you the list of, suppose we have a protein target and you have a list of small molecule. It can give you the binding affinity, right, in numeric value. So likewise, we have something called GNINA, which is like graph-based neural networks. It also, suppose you have a protein molecule, you also submit the PDB file there. You also submit the list of molecule. So what it will give you the ranked list. It will give you a ranked list based on a score, which is determined by a deep learning model, right? So it also gives you the rank list, but rather than the binding affinity, its score is defined by a score which is based on AI model, and that is as accurate, many times more accurate compared to the deep the conventional docking. So it does give you the score on basis of which you can decide which particular molecule to take into account. So you can rank all the things. So it will give you output like that. I hope it answer or something else.
Yeah, yes, sir. But can you suggest any deep learning models which is available, a tool online, so that we can try and experiment on that one?
So GNINA is that, GNINA.
So what it is, sir? G?
Yeah, I will type in the chat. So G.
Yes, sir. Okay, sir. Thank you so much, sir.
Yeah.
Vanita, can you proceed further?
Okay. Thank you, Dr. Bipin Singh, sir, for taking out your time and sparing with us and giving a very elaborate session on applications of artificial intelligence and machine learning in drug discovery as well as in protein design. So it was excellent, and you have given a lot of inputs in the point of how to design or how to go forward in search of certain models to predict the drugs or the protein so that you can design it. So you have introduced us to some of the neural network and also the tools with which you will be able to do it. So thank you so much, sir. And on behalf of the management, principal, and Department of Biochemistry and Nutrition, we take this opportunity to thank you so much, sir. Thank you, sir.
Thank you very much
Now,
for the invitation.
So shall we take a group photograph? I request everybody to please switch on the.
Ma'am, I'll also post the attendance and feedback link in the chat box.
Yeah,
After
meanwhile
the
we can
photo.
take. Yeah, we'll
After the
take a
photo.
photograph. All the participants, please request you all to switch on your cameras so that we can have a quick photograph. Nobody is switching on. All the participants, please switch on your cameras. Okay, maybe we can just take a quick photograph and wind off. Thank you, sir, once again.
Thank you very much for the invite.
On the
Thank
weekend,
you.
you have. You can stop recording, I think, Vanita. I think on the weekend you have taken your time and spared your weekend time for us, so that's really